Students, further in continuation with the earlier, the vascular system, primary phloem. We will study primary phloem. Primary phloem consists of protophlloem and metaphloem, both recognized on the basic of differentiation. The first form phloem elements are called protophlloem, while later form are called metaphloem. The elements of protophlloem are slender and elongated with their cell wall made up of cellulose, as these develop under stress due to rapid elongation of the organ. The metaphlloem element differentiate after the elongation phase of the organ is completed, so these are not subjected to elongation. The protophlloem consists of sieve tube in angiosperm and sieve element in gymnosperm and pteridophyte. Companion cells are rare or altogether absent. Metaphloem elements are complex in nature and consist of sieve tube or cells, companion cell, parenchyma and fibers. In monocot and in certain dicots such as cucurbita, the primary phloem remain persistent throughout the life of the plant organ, as in secondary growth, and rarely found in them, and hence secondary phloem is not developed. In other case where secondary growth is found, it takes the function of primary phloem, which often get crushed. The phloem is usually found on the outer side external of the xylem of the vascular bundle in the stem, and on the adaxial side in case of leaf and leaf-like organ. But in the member of Solanaceae, Cucurbitaceae, Compositae, Apocynaceae, Asclepiadaceae, Convolvulaceae of the dicotyledons, a part of the phloem may be internal in addition to external. The internal phloem is present on the inner side of xylem and is normally called intraxillary phloem. Both type of phloem are structurally similar, but the internal phloem differentiate later than the external phloem. Yet another type of phloem, often referred to as interxillary phloem or included phloem, is found in the member of family like Loganeaceae, Acanthaceae, Combritaceae, etc. But it is a secondary phloem formed due to the abnormal activity of the cambium. Cambium occur in the form of a thin strip of cell between xylem and phloem in the vascular bundle of the stem of dicot and gymnosperm. It is a part of the procambium or primary meristem and is classified as a lateral meristem occurring parallel to the plant axis. It consists of two types of cells: the elongated cell with the tapering end called the fusiform cell, and second is the small, more or less isodiametric cell called the ray cell. The cambium by its meristematic activity produce secondary tissues and brings about secondary growth. The fusion initial form the secondary xylem and the secondary phloem, whereas ray initial form the ray cell found in the xylem and phloem, which occur in transfer or horizontal series. The cambium consists of one to few layers of rectangular or roughly brick-shaped cell. The cell wall is thin and cellulosic in nature, which surround highly vacuolated cytoplasm with a large nuclei. The cell wall often contain primary pit fill. Transfusion tissues. Transfusion tissues are found in leaves of certain gymnosperm, Cycas and Pinus. These are the special type of conducting tissues found present around the vascular region. In Pinus needle, these are of two types: the transfusion parenchyma cell and the transfusion trachees. Transfusion parenchyma are the thin wall, non-lignified, contain dense cytoplasm and prominent nucleus. These frequently occur near the phloem region. Transfusion tracheae are short, tracheid-like cell with a lignified wall, bordered pit, scleriform or reticulate thickening. These do not contain protoplasm. Hence, non-living and are found adjacent to the xylem. The nature of the origin of transfusion tissue is disputed. Some believe that them to be derived from the centripetal xylem, while others consider them to as a metamorphosis of the parenchyma located outside the vascular region. Their function is also uncertain. Probably these translocate water or food substance from the mesophyll to the vascular bundle or vice versa. Some consider them as a water storage tissue. Thank you.